So my name is Ploingarm Petsophonsakul and I'm representing ESR 11. My project title is Nutraceutical Modulating the Vascular Vitamin K System, and Maastricht will be my home base for the next few years. So let me tell you a little bit why I have chosen this topic. As some of you might already know where I come from, I am originally from Thailand. And in Thailand, food is culture. We love to cook, we love to eat, and we love to talk about our food. As for me, I like spending my weekends in the kitchen experimenting. I like to create healthy food, and nutrition is my passion. So I grew up in a family with all sorts of medical history, with my grandma having hypertension, my uncle had diabetes, so I'm feeling that it's very common and I'm pretty used to it. Not until one day that my dad, who is a healthy guy, physically active, at the age of 50, got a sudden heart attack. Luckily, he was a physician himself, so as soon as he felt the chest pain, he immediately admitted himself to the emergency room and got himself operated on time. So today, he's still alive, still doing surgeries to his patient. But this always makes me wonder, what would be the outcome if this would have happened to the other members of my family? So we know that cardiovascular disease is multifactorial, and if there's anything that we can do to hold or slow down the progression, that would definitely make such a huge impact on the patient's quality of life. So I got very excited when I learned about the EU Horizon 2020 Evolution project. This I got from the website. It says it's an innovative concept of boosting natural protective mechanisms operating in our body. And that for me was really intriguing. The project Nutraceutical Modulating the Vascular Vitamin K System has nicely executed the intention of evolution in using the endogenous mechanism, vitamin K system, which is already in our body to protect us against cardiovascular disease. So this project has perfectly combines all my interest in using the nutritional strategy, in the disease, and also in being proactive in preventing the disease. So let's talk a little bit more about what my project is all about. So cardiovascular disease has been described in countless literature in the context of endothelial dysfunction, lipid infiltration, and inflammation. But however, if we have a look at the vasculature, the majority of cell type which can be found there is actually the vascular smooth muscle cells. So vascular smooth muscle cells is essential for a good performance of the vasculature. So by contracting and relaxing, it alters the luminal diameter and enables that the vessel maintains an appropriate blood flow. So vascular smooth muscle cells also involves in many pathologies, for example, in atherosclerosis and also in aneurysm formation. So my focus is in the vascular smooth muscle cells in aneurysm formation, and with that I will come and talk about it later. So in these pathologies, vascular smooth muscle cells plays an important role in vascular remodeling. The contractile vascular smooth muscle cells can switch itself to a synthetic phenotype and secrete abundant protein vesicles, which then later mediate the vascular remodeling. Vascular smooth muscle cells also synthesize a vitamin K dependent protein, which helps regulate this so-called phenotypic switching, and this vitamin K dependent protein is the MGP. So MGP helps maintain the contractility of the vascular smooth muscle cells by binding to the BMP, which then blocks the further downregulation of vascular smooth muscle cells, differentiating towards an osteochondrogenic phenotype and thereby inhibit calcification. MGP also prevents the mineralization of the protein vesicles, so lacking of MGP results in vascular calcification. And since MGP has to be carboxylated by the vitamin K in order to become functionally active, the status of vitamin K becomes very crucial. So vitamin K is extremely important throughout our life. At least in the Western world, every newborn child receives a vitamin K shots to prevent the development of brain hemorrhagic disease. Of course, vitamin K is needed to activate the coagulation factor. And in later life, vitamin K, as I already mentioned, is needed to carboxylate the MGP and therefore preventing vascular calcification. So let's talk a little bit more about vitamin K. So vitamin K in nature can be found in two forms: the vitamin K1 phylloquinone and vitamin K2 menaquinone, with an important derivative, long-chain menaquinone 7. So both vitamin K1 and vitamin K2 are equally active when it comes to carboxylating the vitamin K-dependent protein. However, it is the vitamin K2 which activates the vitamin K-dependent protein in the vasculature and also having a better bioavailability. So vitamin K2 seems to be more powerful. And many species already realize the importance of vitamin K. Vitamin K1 phylloquinone can be found in green leafy vegetables, while vitamin K2 is actually synthesized by the bacteria. So this friend here already knows where he can find the nutritious MK7 from, but what about us? I hope we're not going to do this. So as for us, there is this Japanese food product, which I believe it's the third time already everyone is hearing about. It's the famous natto, famous for being rich in MK7 and also for being very difficult to consume. Luckily, we also have a supplementation form of MK7 available right now, which makes everything so much easier. So apart from the role of vitamin K in the carboxylation cycle, activating MGP, the calcium metabolism, we believe that vitamin K has certainly more than just one function. So vitamin K2 is synthesized by the bacteria in order to generate ATP, and this is most likely what is happening in our cells as well. So the vitamin K redox itself intrinsically functions as a free radical scavenger and thereby posing an antioxidative properties, so reducing the oxidative stress. And as we know, oxidative stress is the main driver of the vascular smooth muscle cells in cardiovascular disease pathologies. And therefore, it is this role of vitamin K that I would like to investigate. So vitamin K1 and K2 will be compared with a special focus on MK7 in collaboration with Natto Pharma. And one of our focus of vitamin K metabolism in cardiovascular disease will be in aneurysm formation. And aneurysm is a dilation of the vessel walls greater than 1.5 diameters. It is caused by weakening of the vessel wall and results in this balloon-like vessel. So an untreated aneurysm will only lead to an expansive progression, rupture, hemorrhage, and finally death. With smoking being one of the strongest modifiable risk factor. However, genetic disease like Marfan syndromes, these patients, they are also subjected to aneurysm formation. If we have a look at the histological level, aneurysm is characterized by pronounced oxidative stress, vascular smooth muscle cells phenotypic switching, apoptosis, and all of which leads to an extracellular matrix degradation, and thereby weakening of the vessel wall. And if you remember earlier, the vascular smooth muscle cells phenotypic switching results in calcification, and calcification or aortic stiffening will only lead to a more accelerating dilation and aneurysm growth. So what is the role of vitamin K here? Vitamin K reduces oxidative stress. It activates the MGP in order to prevent vascular calcification. So it is here and here that it would like to modulate this process with vitamin K. So if you have a look at the outline of my research project, it will comprise in vitro work. Our lab is having expertise in vascular smooth muscle cells. We are working with primary human vascular smooth muscle cells from both healthy donor and also aneurysm samples. And the cells were isolated by ourselves, and which we can also modulate to different phenotypes at any time that we want. We're also using the biohybrid screening system that we can screen oxidative stress from both the vascular smooth muscle cell samples that we have. And as I already mentioned earlier, I will be testing the effect of vitamin K derivatives, K1, K2, MK7. Nevertheless, we're also open and be interested into looking at the novel or synthetic vitamin K derivative, which may enhance the potential of vitamin K. In the preclinical setting, we also have very nice mouse models, which we can induce apoptosis and also study the phenotypic switching. The first one is the DT mouse, which is a transgenic mouse expressing human diphtheria toxin receptor from the SM22 promoter. The idea is that if you injected the diphtheria toxin, it will induce the apoptosis of vascular smooth muscle cells and also reduces the cellularity up to 70%. Therefore, we can manipulate and also investigate apoptosis in related to aneurysm formation. And another mouse is the smoothulin knockout mouse. The smoothulin mouse is lacking smoothulin B gene, which is one of the strongest marker for contractility. So lacking of smoothulin, the smoothulin B gene is taken out and replaced by a cherry construct, and thereby if there is a loss of red signal, it indicates the loss of vascular smooth muscle cells contractility. So also the cardiovascular of the smoothulin mouse has been described in the literature that it is similar to those patients who have established hypertension. So therefore, it is even more attractive as a mouse model to study. Additionally, we also have access to the biobank of aneurysm patients, in which we receive both tissue and plasma samples that we can instantly measure the status of vitamin K and MGP right away. So, and this is it for my project. I hope you enjoy it and be excited for this project as much as I am. And for now, just let the lab be my kitchen, and I hope to deliver a good dishes for the next time we meet. Special thanks for my supervisor, Leon Scherges, and the rest of the team, and also John Paul and Annabelle Scott. Thank you very much.